Right now, if you are training consistently and not seeing the results you expected, the most likely explanation has nothing to do with your program, your effort, or your sleep. It has to do with three specific molecular signals that your current food choices are almost certainly not delivering in the right amount at the right time through the right biological pathway, not across the day in each individual meal. Because the switch that determines whether the protein in your food becomes new muscle tissue or gets oxidized for energy does not respond to total daily protein. It responds to the specific molecular trigger that arrives at the muscle cell in a single eating occasion. And if that trigger does not reach its threshold, the signal does not fire. The effort was real. The instruction to build was never sent. This is the mechanism most people training hard and eating enough protein have never been given, and it explains with precision why someone can eat 150 grams of protein per day, train 4 times per week, sleep 7 hours, and still be looking almost identical at the end of the year to how they looked at the start. Here is the mechanism that makes everything else make sense. Inside every muscle fiber lives a molecular switch called mTOR1, the mechanistic target of rapamycin complex 1. mTOR1 is the master regulator of muscle protein synthesis. When it is active, the ribosomes inside the muscle cell begin translating messenger RNA into new contractile protein. When it is suppressed, that translation process stops regardless of how much raw material is available. Think of mTOR1 as a construction foreman standing on a building site. When the foreman is dormant, nothing gets built. Protein arrives, the workers stand around, bricks pile up on the site. But when the foreman receives the right activation signal, the entire site responds. Scaffolding rises, steel beams connect, new muscle protein is synthesized at the rate your training is demanding. The problem is that mTOR1 does not wake up for just any food. It has a specific activation threshold. The foreman does not respond to a general delivery of construction material. He responds to one specific alarm clock, and that alarm clock is a single amino acid. Research confirmed by Rivera Bermudez and colleagues in 2025 established that unless your meal delivers enough of that specific amino acid at sufficient plasma concentration, the foreman stays asleep regardless of how much total protein you consumed across that day. Below the threshold, the signal does not fire. At and above it, muscle protein synthesis activates fully and the construction begins. Most people eating enough total protein are still missing the signal in individual meals because they do not know what the alarm clock is, what the threshold is, or which foods reach it most reliably. Here is food number 1, and it is already sitting in your kitchen right now: whole eggs. Not for the protein grams, for leucine. Leucine is the specific amino acid that functions as the alarm clock for mTOR1. It activates mTOR1 through the cestrin-2 and gator-2 sensing complex. The mechanism works like this. In the absence of sufficient leucine, cestrin-2 binds and inhibits the gator-2 complex, keeping mTOR1 away from the lysosomal membrane where it must dock to reach full catalytic activity. When plasma leucine rises above the activation threshold, leucine binds cestrin-2, releases the inhibition. RAG-GTPases shuttle mTOR1 to the lysosomal surface, and the full muscle protein synthesis cascade activates. Without a sufficient leucine spike in a single meal, mTOR1 stays below full activation regardless of total daily protein intake because the inhibitory grip of cestrin-2 is never fully released. The threshold is approximately 2.5 to 3.0 grams of leucine in a single meal. This is established across Moore and colleagues in 2009, Whithard and colleagues in 2014, and confirmed in the 2025 review literature. Below 2.5 grams of leucine in a single eating occasion, muscle protein synthesis increases only modestly. At and above it, the response is near maximal. Adding additional leucine beyond the threshold does not produce additional MPS because the pathway is already fully activated. 3 whole eggs deliver approximately 1.65 grams of leucine. 4 whole eggs cross 2.2 grams. Combined with any other leucine-containing food in that meal, a meal built around 3 to 4 whole eggs reliably reaches the activation threshold. Below it, the foreman stays asleep regardless of how much protein arrived at the site. But here is the finding that changes how you think about the egg specifically. Research confirming that whole eggs produce greater muscle protein synthesis than an equivalent protein amount from egg whites alone establishes that the yolk contains compounds that amplify the anabolic response beyond what the leucine alone explains. The yolk contains phosphatidic acid, which directly activates mTOR1 through the phospholipase D signaling pathway independent of leucine. It contains the cholesterol precursors that serve as raw material for testosterone synthesis in the Leydig cells. And it contains vitamin D, which research has confirmed plays a direct role in muscle satellite cell function and the expression of muscle-specific genes that determine the capacity of the muscle to respond to both training and nutrition. Think of the whole egg as a compound signal. The leucine wakes the foreman. The phosphatidic acid from the yolk sends additional instructions directly to the construction supervisor's inbox through a second pathway that does not require the leucine route at all. Both signals arriving simultaneously produce a larger and more sustained construction response than either signal arriving alone. The specific failure mode most people are committing, eating 3 egg whites and discarding the yolks. In doing this, they are reducing the leucine content below the threshold, removing the phosphatidic acid that activates the secondary mTOR1 pathway, removing the cholesterol substrate that Leydig cells need for testosterone synthesis, and removing the vitamin D that supports satellite cell function. All 4 molecular advantages of the whole egg are in the yolk. The white contributes the structural protein. The yolk carries the biological instruction. Discarding it because of a 30-year-old hypothesis about dietary cholesterol and cardiovascular risk that the published evidence has not supported in the specific population of people eating eggs as part of a whole food diet is the nutritional equivalent of buying a car and throwing away the engine. Here is food number 2, and this is where most people plateau without understanding why. Wild-caught fatty fish, specifically salmon, mackerel, or sardines. EPA and DHA, the omega-3 fatty acids found at highest concentration in cold water fatty fish, address the problem that prevents the leucine signal from producing its maximum effect in most people who are training consistently. That problem is anabolic resistance. Anabolic resistance is the state in which the muscle protein synthesis pathway responds to leucine and training with less output than it would under optimal conditions. It is the reason 2 people can eat the same protein, do the same training, and produce different amounts of new muscle tissue per week. The difference between them is often the inflammatory and membrane environment in which their mTOR1 signal is trying to operate. EPA and DHA have been confirmed across multiple published research programs, including the landmark Smith and colleagues studies at Washington University, to increase the sensitivity of skeletal muscle to anabolic signals. The mechanism operates through 2 independent pathways simultaneously. The first is membrane fluidity. EPA and DHA are incorporated directly into the phospholipid bilayer of the muscle cell membrane over 4 to 8 weeks of consistent dietary intake. Their incorporation increases membrane fluidity, which improves the efficiency with which transmembrane signaling proteins, including the insulin receptor, the mTOR complex scaffolding proteins, and the downstream signaling cascade, operate. Think of the muscle cell membrane as a lock mechanism. Leucine is the key, but the precision of the lock determines how completely the key opens the door. A rigid omega-6 dominant membrane is a stiff, poorly machined lock. An EPA and DHA-enriched membrane is a precisely engineered lock that opens fully when the leucine key arrives. 8 weeks of omega-3 supplementation in the Smith studies produced significantly greater muscle protein synthesis rates in response to the same amino acid and insulin infusion compared to the pre-supplementation baseline. The leucine dose did not change, the membrane environment did, and the output changed with it. The second mechanism is anti-inflammatory resolution. Every resistance training session produces a local inflammatory response in working muscle tissue through elevated IL-6, TNF-alpha, and prostaglandins synthesized from the arachidonic acid in the omega-6 dominant cell membranes of most people eating a standard Western diet. This acute inflammation is normal and necessary for the initial muscle repair signal. But when it fails to resolve promptly and becomes chronic, low-grade exercise-induced inflammation, it creates a state of anabolic resistance in which the mTOR C1 response to leucine is blunted and the satellite cell activation required for muscle hypertrophy is suppressed. EPA and DHA are the precursors to the specialized pro-resolving mediators, resolvins, protectins, and maricins that terminate the inflammatory cascade and restore the tissue to an anabolically sensitive state. 3 to 4 servings of fatty fish per week, or 2 to 3 grams of combined EPA and DHA daily from a verified source, consistently replaces the omega-6-derived inflammatory mediators in the muscle cell membrane with the omega-3-derived resolution mediators that end the inflammation before it becomes chronic. A 2025 RCT published in PMC on omega-3 combined with resistance training confirmed an 11.46% increase in counter-movement jump performance alongside measurably increased muscle cell anabolic sensitivity in the omega-3 group compared to the resistance training plus placebo group. Same training, different membrane environment, different output. The specific failure mode most people are committing: eating chicken breast 5 times per week and salmon once. Chicken provides excellent leucine. It provides almost no EPA or DHA. A diet built primarily around chicken, eggs, and plant protein sources without consistent fatty fish or concentrated omega-3 supplementation is a diet that delivers the alarm clock reliably but never improves the quality of the lock it is trying to open. The foreman wakes up. The instructions he sends are transmitted through a poorly maintained communication system. The construction that results is measurably less than the same alarm clock would produce in a better maintained building. Here is food number 3, and this is the mechanism the fitness industry has barely begun to explain. Full-fat plain Greek yogurt with berries, specifically for the gut-muscle axis. The gut-muscle axis is the bidirectional communication pathway between the gut microbiome and skeletal muscle tissue, and it is one of the most significant and most underreported determinants of how efficiently dietary protein is converted into new muscle tissue. Research published in Frontiers in Microbiology in 2025 confirmed that short-chain fatty acids produced by gut bacteria through the anaerobic fermentation of dietary fiber, primarily acetate, propionate, and butyrate, are absorbed from the gut lumen, enter systemic circulation, and activate the mTOR P70S6K signaling pathway directly in skeletal muscle tissue. Butyrate specifically activates mTOR and simultaneously inhibits the FOXO-mediated ubiquitin proteasome degradation pathway that breaks down existing muscle protein. It is producing a net anabolic signal in muscle tissue from the gut independent of dietary protein intake through the SCFA signaling route that most nutrition science has not yet communicated to the fitness audience. The 2025 Frontiers in Microbiology, Sarcopenia, and Gut Muscle Axis review confirmed that age-related declines in SCFA-producing bacteria, specifically Faecalibacterium prausnitzi, Roseburia, and Akkermansia muciniphila, are directly associated with a shift toward muscle protein breakdown that characterizes the progressive sarcopenia of aging. And restoring SCFA levels through dietary fiber and fermented food intervention reverses those trends by restoring the gut-muscle axis anabolic signaling that the microbiome decline was suppressing. Full-fat Greek yogurt addresses this through three simultaneous mechanisms: the live probiotic cultures, Lactobacillus bulgaricus and Streptococcus thermophilus in all commercial Greek yogurts, plus additional strains in live culture products, directly improve the relative abundance of SCFA-producing bacteria in the gut microbiome with consistent daily intake. The casein protein in Greek yogurt provides a slow-release amino acid profile that sustains plasma leucine elevation across 3 to 7 hours, compared to the 1 to 2 hour peak of fast-digesting whey, producing a more extended mTOR activation window that is particularly relevant when this meal is consumed before sleep and the muscle protein synthesis program needs to run across the full overnight recovery period. And the fat content of full-fat Greek yogurt slows gastric emptying, moderating the amino acid delivery rate and extending the leucine availability window further than the same casein in a low-fat product. The berries are not a garnish. Blueberries, blackberries, and raspberries contain anthocyanins and other polyphenols that serve as prebiotic substrate for Akkermansia muciniphila and bifidobacterium strains in the colon. Research confirmed that consistent berry intake significantly increases the relative abundance of these SCFA-producing species over weeks of regular consumption. The berries are feeding the gut microbiome that is feeding the muscle. And the anthocyanin anti-inflammatory effect produces the same resolution of exercise-induced anabolic resistance that EPA and DHA provide through their own pathway, meaning berries and fatty fish are addressing the same anabolic resistance problem through two independent routes simultaneously. The specific failure mode most people are committing: eating low-fat Greek yogurt and adding sweetened granola or fruit juice on top. Low-fat Greek yogurt removes the fat that slows gastric emptying and extends the casein-leucine window. Sweetened granola adds refined carbohydrate that produces an insulin spike, raises mTOR through the glucose-insulin pathway, and simultaneously activates the de novo lipogenesis program that competes with muscle protein synthesis for the cellular resources that mTOR directs. And fruit juice adds fructose that goes directly to hepatic de novo lipogenesis through the unregulated pathway described in the triglyceride script in this series. The combination of low-fat yogurt plus sweetened granola plus juice is delivering some leucine, disrupting the casein window, and adding two mechanisms that partially redirect the mTOR signal away from muscle building. Here is the honest framing. The leucine threshold research from Moore, Wittard, and Rivera-Bermudez is published across Cell Metabolism, the American Journal of Clinical Nutrition, and the Journal of Physiology. The Cestrin-2 Gator-2 mTOR-1 mechanism is established molecular biology, confirmed across multiple independent research groups. The HMU Smith-Washington University omega-3 and muscle protein synthesis augmentation studies are published in the Journal of Physiology. The 2025 PMC RCT on omega-3 and resistance training is published. The Frontiers in Microbiology 2025 Gut Muscle Axis and SCFA research is published and peer-reviewed. The phosphatidic acid and secondary mTOR-1 activation from egg yolk is published in the Journal of the International Society of Sports Nutrition. The honest caveat is this: the three foods described in this script address three specific and independently documented molecular pathways to mTOR-1 activation and anabolic sensitivity. They do not replace adequate total daily protein intake. They do not replace progressive resistance training. They do not address sleep, hormonal status, and energy balance, which are the other major determinants of muscle protein synthesis rates. What they do is ensure that the protein you are already eating sends the strongest possible molecular signal to mTOR1 through the leucine threshold pathway, through the optimal membrane environment that omega-3s create, and through the gut-muscle axis SCFA signaling that most people's microbiome is failing to produce adequately. Here is the complete picture. 3 whole eggs with yolks provide the leucine that crosses the mTOR1 activation threshold through the cestrin-2 pathway, and the phosphatidic acid that simultaneously activates mTOR1 through a second independent route. Wild-caught fatty fish consumed 3 to 4 times per week incorporates EPA and DHA into the muscle cell membrane, improving the lock quality through which the leucine key operates, and provides the resolution mediator precursors that resolve the chronic exercise-induced inflammation creating anabolic resistance. Full-fat Greek yogurt with berries delivers live probiotics that restore SCFA-producing bacteria, slow-release casein that extends the overnight leucine window, and polyphenol prebiotics that feed the gut microbiome producing the SCFA signals that activate mTOR1 through the gut-muscle axis, while simultaneously reducing the inflammatory anabolic resistance that fatty fish is also addressing through its own resolution mediator pathway. 3 independent pathways to the same molecular switch. The foreman wakes up from the leucine. His instructions are transmitted more completely through the EPA and DHA membrane, and the gut microbiome sends additional construction orders through the basement entrance that the SCFA pathway opens. All 3 arriving together produce a compounding anabolic environment that any single food source or single molecular pathway cannot generate alone. Drop a comment right now. Were you eating egg whites and discarding the yolks? Or eating chicken every day without fatty fish? Did you know the gut-muscle axis was a real and documented mechanism affecting your muscle protein synthesis rates? I read every single one. If this changed how you think about why your training effort has not been producing the results the effort deserves, hit subscribe. Every week on the Rewired, we decode the mechanisms behind the habits that determine your health, your brain, and how you age. New mechanism every week. The foreman has always been on the site. The alarm clock, the lock quality, and the basement entrance were waiting for you to use them.